administrators from our RNA centers. For us, this is Liz Leverins. We wouldn't be able to do this without Liz's support, so thank you, Liz. So Clara Kielkopf is our first speaker today. She's a professor of biochemistry and biophysics at the University of Rochester. She earned her BS degree at the University of Wisconsin at Madison and earned her PhD at Caltech. She then postdoced at Rockefeller with Stephen Burley. She started her independent career at Johns Hopkins and then moved to the University of Rochester. One focus of her lab has been solving X-ray structures of U2AF65, recognizing splice site signals. Today, her talk is Targeting U2AF, a vulnerable rheostat of pre-mRNA splicing. And before she starts, I want to remind you that questions can be posted in the Q&A, and I can read them out loud to Clara, or you can use the raise hand feature, and then I can call on you to speak to the audience. And Clara, we're ready for you to take it away.
All right, hopefully you can see the slides now. All right, so good afternoon, everyone. Sure, a few of you are sleep deprived like us after the exciting election, which is still ongoing. So today I'd like to tell you about some new exciting results on targeting U2AF, which is an early stage splicing factor. And I think the interesting take home from this is that we can actually stall splicing by enhancing a U2AF RNA complex. So, and this could be a general way of inhibiting the many ATP-dependent and tightly regulated processes in the cell, is to actually enhance an early checkpoint. All right, as many of you know, the spliceosome is an incredibly complicated and dynamic molecular machine. The pre-messenger RNAs are spliced during rearrangements, sequential rearrangements, among more than 100 proteins and 5 small nuclear RNAs. So it's actually no surprise that splicing is often dysregulated in cancers. It's long been known that there are many, more than 15,000 cancer-associated variants of transcripts that are dysregulated in cancers, including some very prominent kinases and proto-oncogenes. More recently, through genome-wide sequencing and RNA-seq, it's been shown that a lot of these splicing alterations are associated with alterations of the splicing factors themselves. For example, SO3B1, one of the early stage splicing factors, is amplified in many different types of cancers. And SRSF2 is also upregulated and in some cases downregulated in many different cancers. In about 2011, it was also shown that the splicing factors acquire specific point mutations. These occur particularly in hematologic malignancies. For example, in myelodysplastic syndrome, it's thought that, in fact, the majority, more than half of all the acquired mutations that are associated with MDS occur in splicing factors. And these are very interesting mutations because they recur at particular hotspots. They aren't just randomly distributed, and they're rarely null. They're usually a very specific missense mutation that seems to confer a new splicing function or really alter the function of the splicing factor. So what are these myelodysplastic syndromes that acquire these splicing factor mutations so commonly? Well, they're a disease of the bone marrow, which may be why it's related to splicing, is that the bone marrow is constantly generating these blood cells. So in myelodysplastic syndrome, the bone marrow often has a morphological change, as shown here, and this disrupts the blood cell production. It's most common in the elderly. In fact, it's thought to be the sort of the disease of the Trumps, of the people who like to go golfing, and they get a lot of exposure to maybe herbicides on the grass and sun and environmental insults, because these are, of course, acquired mutations. And, but interestingly enough, the poster child for MDS is Robin Roberts, who is an African-American woman. And she was thought to have gotten MDS through chemotherapy for breast cancer. Death from MDS usually results from the lack of blood cells, not from the bone marrow itself. It's just a secondary effect, but of course a major one, of not being able to produce enough healthy blood cells. And currently there's no curative chemotherapy, hence the motivation for this project is there's no chemotherapy that's known to work for MDS patients. Currently, the only curative treatment is a bone marrow transplant, which can be risky for these elderly patients. So this motivates our study, which is to find small molecules that target the early stage splicing factor. In this case, we have focused on U2AF. But to give a little background, one of the most clinically advanced small molecule inhibitors of splicing are a class of inhibitors that universally target the splicing factor SF3B1, shown here. And there are many variants of these. They are naturally occurring small molecules, complicated, as shown down here, and they differ in their general pharmacophores. However, they universally target the same site on SF3B1, which is a binding pocket for the nucleophile of the splicing reaction, the branch site adenosine, which SF3B1 recognizes and protects in an A stage of the spliceosome checkpoint. So interestingly, these have been optimized and from the natural products and have been put into phase 1 clinical trials and have met with some success. So these small molecules will selectively kill cancer-relevant cell lines, such as those that express the MDS-associated mutant splicing factors, and also those that, for example, have transcriptional overload, which is thought to overload the spliceosome itself, through overexpression, hyperactivation of the MYC transcription factor. So there's success here, which has broad therapeutic potential for cancers. And in phase 1 clinical trials in leukemias, which were started in 2017, the optimized SF3B1 inhibitor showed some success, and 15% of the treated patients became transfusion independent. So this is a favorable response, although they were not cured. So there's still a ways to go here. But nothing's perfect. There were some challenges in the clinical trials. I have heard that there's unwanted hair growth, which maybe if you're faced with a life or death treatment, that's okay. But you still would prefer not to have that. And there are challenging to make. The chemical structure has 9 chiral centers, so those have to be enantiomerically pure. And of course, they're limited at this point to one target, although there are other strategies, of course, in the pipeline, for example, SR protein kinase inhibitors and antisense oligonucleotides. So given that there are more than 100 splicing factors, we have a rich, uncharted plethora of potential targets for new therapeutics for MDS. And our laboratory has been working on U2AF for many, many years, but this is a new, exciting opportunity to see what we can do with the observations that we have to date. And so we decided to use the tools in our molecular toolbox in lab to try to target the U2AF, which is a heterodimer here of 2 subunits, U2AF1 and U2AF2. So our rationale was really, as I've said, the successful precedent of the SF3B inhibitors. And the U2AF, it binds in the early stage like SF3B1. In fact, it even interacts with SF3B1 in the A stage of the spliceosome complex. And it's a key checkpoint factor. So by blocking U2AF, one would hope to be able to inhibit the splicing process at an early stage and possibly have some action with SF3B1 inhibitors. Also, the U2AF1 subunit is recurrently mutated in myelodysplastic syndromes itself. It carries an S34F mutation in almost 15% of MDS patients. So that creates a vulnerability that then can be targeted. So in our screen, we used a simple polarization assay. However, this has an advantage in that we know what we're targeting. We have a purified protein complex. The SF1 is an accessory subunit in the first stage before SF3B1. SF1 with the U2AF heterodimer, and we included the S34F mutation that is associated with MDS in hopes of selectively targeting that mutation. So we have a purified protein complex, a fluorescein labeled pre-messenger RNA. Well, it's an RNA splice site. And for our assay, we chose to use the DEK proto-oncogene splice site, which is known to be dysregulated in MDS and sensitive to the S34F mutation of U2AF. We successfully implemented the screen, and we have a nice difference in signal between our controls and our protein complex. First, we screened for inhibitors of our U2AF complex. And we screened a moderately sized diversity library, which is available from the NCI if anyone is interested. But this was a nice size for our ability to purify the protein complex. And it has structurally diverse pharmacophores, which set the stage for subsequent optimization. So if we get a hit, we can probably use our structural knowledge to diversify that hit and screen for a higher potency lead compound. So we completed the screen. First, we looked for inhibitors and then for enhancers. So in our inhibitor screen, we identified 3 compounds that reduced the polarization of the U2AF RNA complex and not of the RNA alone. However, in secondary assays, we found that these compounds bound the RNA, and one of them was elliptoseed, which is a well-known RNA or nucleic acid intercalator. So we were less enthusiastic about these because we thought that probably they were binding in a non-specific way, not specifically to the splice site, which would have been more interesting. And so my student, now Dr. Rakesh Chatriki, who's now working at a pharmaceutical company after a nice postdoc with Kristen Lynch at UPenn, he was working on this project and he said, let's just look for enhancers. I was like, oh no, why would we want an enhancer of a U2AF RNA complex? And he's like, well, you got the S34F mutation, and so maybe you want to enhance the ribonucleoprotein complex in some cases. So I was like, okay, go ahead. So he did the screen on the right, which again it's a statistically significant result as indicated by the Z prime and Z scores. And he identified one compound which reproducibly reduced the polarization of the ribonucleoprotein complex, but not the RNA alone, and had a reasonable EC50 in follow-up assays. Okay, so why should we be excited about this enhancer of the U2AF RNA complex? Well, okay, so he tried next an in vitro splicing assay, shown here. This is an assay of a prototypical ADML substrate, which of course many of you have used many times, in Helin nuclear extract, analyzed by quantitative real-time PCR. And we were quite surprised but pleased that the small molecule did inhibit the production of the spliced product, despite the apparent enhancement of the U2AF RNA complex in our polarization assay. We next reached out to Melissa Jurica at UC Santa Cruz, who is very adept at running these, what I understand are technically challenging, Lab hasn't really done them before, gels on which the spliced products are separated. And she reproduced our result first, that adding the compound would inhibit splicing. So as the compound is added, less and less of the spliced product is formed. And then interesting, she was able to do a spliceosome assembly assay, shown here on the right. And what she found is that our small molecule would stall splicing at the early U2AF dependent stage, the H and E complex. So this showed that yes, it was acting by enhancing and sort of trapping this U2AF RNA complex, but that stopped splicing because you couldn't get rid of U2AF. U2AF has to leave for splicing to proceed to the catalytic stages. So that was an interesting result that could be broadly applied to other multi-stage processes such as this one. So our take home is that trapping a checkpoint complex is a mechanistic strategy for macromolecular inhibition. All right, so then we went on with the details of, okay, what are we really targeting here? We had screened against an SF1 U2AF2 U2AF1 complex with the S34F mutation. The U2F heterodimer is difficult to purify independently, at least for the U2F1 subunit. So we did what we could, and we compared the IC50s of the activity of the compound against the different components that we could make. And we found that the U2F RNA, although certainly less striking, it almost looks cooperative in its activity. It does have a response that is, well, winds up with an EC50 that's very similar to the intact complex. So it did appear the compound is targeting U2AF2, although having the other subunits there enhanced the all or none response. We then used Biochemistry to confirm this with an orthogonal assay. Now, Biochemistry is a strategy in which we are immobilizing the protein molecule on a sensor chip and then flowing. In this case, we're using a GST tag and an anti-GST antibody, and flowing the small molecule across the chip in comparison with a tightly matched DMSO control, because DMSO also will give you a response on this chip. So this is a very sort of tricky assay because the response of the small molecule should be very, very low. If you see any more than an apparent one-to-one complex, then you may be looking at precipitation of the compound on the sensor chip. But what we saw is that the compound does seem to bind to the U2F subunit alone. In this case, we were not able to include RNA, so that the parent affinities, of course, are much lower than they would be for the ribonuclear protein complex. We then compared different constructs, including the isolated RNRM, or RNA recognition motif domains of U2AF2, as well as a construct in which the inter-RM linker has been shortened. And we only saw a reasonable binding response when the intact RNA binding domain was used in this assay. So we concluded that somehow the arrangement of the RMs was most likely important for binding of the small molecule. Next, we reached out to our neighbors, Ram Samadrala and his student, Zach Falls at University at Buffalo, and asked if they could dock our compound against our structures of U2F2. Of course, we tried to crystallize it, and we were not able to grow crystals in the presence of DMSO, so that was always a challenge. But the docking suggested, in agreement with the binding assays, that the compound binds in between the two RMs. We then introduced mutations and confirmed that, I'm sorry, I should have updated this slide. And we then introduced mutations at this site that confirmed the compound binds between the two RMs of U2F2. So we're pretty confident of that. So in a final step of investigating the structure-activity relationships, we took a small step in that direction by comparing the activities of 6 different variants of this compound in vitro splicing assays. And this is at a 50 micromolar addition. And using an NCI obtained compound, we had also independently synthesized the compound, and the in-house synthesized compound was similar or maybe better activity. We saw in these assays that including the negatively charged portion of the molecule was important for its ability to inhibit splicing, but also a hydrophobic portion also needed to be present for the ability to inhibit splicing. And in fact, the shape of that portion needed to be somewhat three-dimensional, or at least have a critical amount present. On the other hand, if we could add a little bit to this hydrophobic portion, we saw a similar response as the parent compound. But nonetheless, this shows that there's a negatively charged part of the molecule and a hydrophobic portion that are somehow linking the two RNs together. So next asks whether, considering that in vitro, the compound would inhibit splicing of the ADML prototype, we asked if treatment of a cell line would result in changes in splicing of representative U2A sensitive transcripts. Now, these transcripts are from the Fu paper in Nature Structural biology from, I think, 2014. I should have put this here. But we were able to reproduce that when treated with control siRNA, there are changes in splicing of these 3 transcripts, as he had seen based on his RNA-seq and CLIP-seq data. And when we treated the cells with increasing amounts of compound, we likewise could reproduce similar changes in splicing at these sites as the Fu lab had seen. So this was satisfying, and it led us to go on to the next step, which is to ask, could this compound exacerbate splicing defects in a relevant, MDS relevant cells? And so for this experiment, we used a CRISPR edited cell, K562 leukemia cells, provided by our collaborators Tim Grobert and Matt Walter at Harvard and Washington University. In this case, we're using the CRISPR edited forms from Tim Grobert, and Matt, as you will see, also provided a different cell line that is inducible, dox-inducible. Here we're using the edited cell lines. And these were either expressing a control wild type U2AF1 or an S34F mutant that had been CRISPR edited. And so we say again that, interesting, of course, the S34F does mutation of U2AF1, which is found in about 15% of MDS, does alter splicing of these representative transcripts compared to the wild type counterpart. And addition of the hit compound will exacerbate these splicing defects. It actually makes it a little bit worse. So this is good news for us that we are able to alter splicing with our compound and that we make worse effects that are seen with an MDS relevant mutation of U2AF1. So then we asked whether treating cell lines that were expressing the S34F mutant, the MDS mutant of U2AF1 with the compound would selectively kill them at lower concentrations than the wild type counterpart. And we tested 2 different cell lines in this case, the CRISPR edited ones, which we used for the RT-PCR experiments, in comparison with an inducible, dox-inducible cell line. And of course, the dox-inducible cell line does express more U2AF1. And so we see that in both cases, there is the S34F expressing cells are killed at lower concentrations of the compound than the wild type counterpart. So the S34F cells, expressing cells, are in red on both plots, and the wild type counterpart is in blue. And we do see that at lower concentrations, there is a greater cell death in counts of the cell surviving fraction. It also may be worth noting that the ones that are U2AF1 inducible appear to be more sensitive to treatment with the compound than those that are edited, suggesting that this higher level of U2AF1 actually confers, again, a greater sensitivity, which holds promise for those cells which are overexpressing splicing factors in other hematologic malignancies. All right, so that is my talk. And so to summarize, our goal was to identify a small molecule modulator of U2AF2 that's a new means to inhibit RNA splicing and potentially in the longer term as a tool to investigate or treat MDS, which we have found a hit, which now is ready to be optimized. And this compound will preferentially kill cells that are expressing an MDS relevant mutation compared to the wild type counterparts. So this is all very promising. But I think really the most interesting, maybe for us or basic lab scientists, is that this is a potentially general strategy for inhibiting macromolecular processes, is that rather than just inhibiting something, you can actually promote an early checkpoint, and that will stall things. So I think that's an interesting and potentially useful take home for future. All right. And I'd like to thank all of you for your attention and the organizers for the opportunity to talk. This work on this compound was started by Dr. Rakesh Shetwicki, who is now at Arrakis Therapeutics, continued by Callan Feeney in this laboratory. in my laboratory who now is, he's going to be a teacher, a high school teacher, and I hope to continue to help his students as well. And Mary Palvino, a very talented technician in the laboratory who did all the cell culture, and Dr. Jenkins, who is the leader in the Biochemistry. He's a great, great biophysicist. All right, thank you for your attention.
Thanks, Clara. Now we have time to take some questions before we move on to Paul. And we have various ways that questions can be asked, either through the question and answer or by raising your hand and we can let you speak. So one question came through in the chat, which is from Gil Oman. And he asks, how do you identify altered pathways downstream from the U2AF S34F mutation, if you do?
Would that be from the S34F mutation itself, not from the hit compound? So many, our collaborators, in fact, there have been many efforts to move in that direction and really relate the S34F mutation to sort of a cause-effect relationship with MDS, which is hard to do. And that's not, so Tim Grobert and Matt Walter have been working on that, and they've done RNA-seq and a variety of other assays to try to really pinpoint that, and it's hard. Most recently, they're very excited by R loops. So they had a paper just a year ago, I think, and the relationship between the S34F mutation, not only it is invokes splicing changes, and Michael Green's laboratory has shown that it also will affect autophagy and polyadenylation and many other processes. And then Matt Walter and Tim Grobert are excited to see that it also shows dysfunctional R loops, which are related to DNA damage and repair. So and then of course, I would like to optimize our hit compound a little bit, but we should do RNA-seq and really see what pathways are going on down there.
So another question came in through question and answer from Chang Yi Yang. And the question is, does your compound affect the IRAK4 splicing?
I don't know, but that's a really interesting question that we should look at. IRAK4 was recently shown, thinking of the literature, is that affected by SO3B1 mutations as well? Yeah, we should go in and look at that. Thank you.
So another question came in through the question answer from Robert Weinberg. And he asks, are there specific genes targeted in MDS by the mutant U2A spliceosomes?
Yes. Yes, I don't know that there's a very strong, DK is one of them, the DEC proto-oncogene. There are only about a few, it's not an overwhelming amount of genes that are altered by this mutation. It's actually only maybe 3% or less by memory. But how those are related to downstream effects is not as clear, not entirely clear. In polyadenylation, the Green lab has shown, Michael Green's lab at UMass Med has shown that ATG7 specifically has a change in polyadenylation that may be related to certain effects. Although I think it's not strictly been shown that that's going to cause MDS. So yes, there are a specific handful of genes, but how those relate to MDS is not so clear. Sorry to not be more helpful there. But.
We'll take one more question. Peng Yao asked, the compound can still kill wild type U2AF expressing cells. Mm-hmm. Does that suggest optimal activity or expression level of U2AF1 is necessary to maintain normal healthy blood cells?
Absolutely. And that also is related to the observation that the splicing factor expression levels and copy numbers are often changed in cancers. But on the other hand, this could be good for us because if cells that have a little bit of a skew in their expression levels of splicing factors are sensitized to U2AF inhibition, then that means that that's a potential application for us, and that broadens the potential number of different cancers that could be treated with a similar optimized compound. So thanks, Pinyao.
So thanks for asking questions. I think it's time that we move on. There are several that are unanswered, but Clara can type some answers in while Paul speaks. So thanks, Clara.
Thank you.
So our second speaker today is Paul Boutz, who's a professor of biochemistry and biophysics at the University of Rochester. Paul earned his BA at Rice University and his PhD at UCLA. He then postdoced at MIT with Phil Sharp. He joined us here in our Center for RNA Biology, but he was also a pioneering hire in our new Center for Biomedical Informatics. At U of R, Paul's been focusing on understanding the processes that shape the transcriptome. Today, he's going to talk about tumors taking the shortcut, CDK12 and CDK13, in early transcription termination and nuclear RNA surveillance.
See the screen okay?
Yes, just fine.
Okay, great. Thanks, Dave, for the introduction. So my group is interested broadly in various types of RNA processing and how that relates to, in particular, to the biology of cancer cells. So we study alternative splicing, but in the last couple of years, one of the focuses that has become really interesting is two genes, CDK12 and CDK13, that really represent the interconnections between transcriptional processes and both RNA processing, but also what happens to RNAs after they're fully mature. So CDK12 and CDK13 are cyclin-dependent kinases. This is a broad family of kinase molecules that are dependent on a partner called a cyclin for their activity. Many of you, I'm sure, are aware of the role of the cell cycle CDKs, which are quite well studied, but there's also a separate branch of the family that are dedicated transcriptional regulators. And CDK12 and 13 belong to this subgroup. So in eukaryotes, as far back as yeast, the RNA Pol2 has this rather long C-terminal domain that consists of these heptapeptide repeats. I'm showing here the consensus for the mammalian repeat, of which there are 52. Not all of them exactly like this, but most of them very similar with a few substitutions at various locations. But what this domain is responsible for is really acting as a platform to integrate transcription, to regulate the transcription cycle, and to bring in other machinery. And so the most well-studied modifications that occur among many are the serine 2 and serine 5 phosphorylations. These are carried out by a number of different cyclin-dependent kinases. And so whereas serine 5 is completely phosphorylated by the CDK7 homologs, even in yeast, the serine 2 kinase activity is really divided between two different homologous proteins. So CDK9 in mammals, as part of the PTEF-B complex, is responsible for serine 2 phosphorylation to release the polymerase into productive elongation. In vertebrates, actually, there are two different CDK12 homologs. And it's been unclear until very recently why that is, why there are multiple kinases that carry out this same modification. And we'll talk a lot more about that during this talk. So as I mentioned, one of the major functions of CT dephosphorylation is in recruitment of other machinery to the polymerase to carry out transcription and RNA processing. And this includes both factors that enable elongation to occur efficiently, particularly through the chromatin environment that the polymerase has to be able to traverse. Additionally, RNA processing machinery, including the spliceosome, as well as the cleavage and polyadenylation machinery, are brought into interaction and through the C-terminal domain. And the dynamic phosphorylation of that domain both promotes different stages of the transcription cycle, but also brings these other factors in and out to be appropriately coordinated. So the project that I'm going to talk about today really started when I started working with an outstanding graduate student in Phil's lab by the name of Sarah Dubry. And we set out to understand at the time what very little was understood about the function of CDK12, and again, why are there multiple serine 2 kinases regulating this process? So we began by genetically depleting it from mouse embryonic stem cells. And using these high resolution chip assays, one of the first things that we found is that, as expected, when CDK12 is depleted, and so here we're looking across gene bodies, a metagene from the transcription start site to the transcription end site, and we saw a depletion, though not a complete depletion, but a reduction in the amount of serine 2 phosphorylation. We also saw this very subtle effect where, in the absence of CDK12, the total polymerase density gradually began to accumulate as you moved more and more towards the 3' end of the gene. And so we suspected that perhaps this was indicative that piling up within the gene body due to a slowing of elongation. But because we didn't have any dynamic assays to really assess processivity and elongation velocity,
So we seem to have a temporary problem here. Hold on while we try to fix this.
Not sure what happened there. I lost my connection. Okay. Here we go. Okay. So the full length isoforms were typically decreased in this case due to the upregulation of early termination events. And so as a result of this, many genes, particularly in components of the homologous repair machinery, which happen to have a lot more of these intronic polyA sites, They tend to be very long genes with many introns. And these sites were typically more sensitive to the loss of CDK12. And so what you can see, or what you can easily discern, is that in a gene with multiple IPA sites, even if there's a small increase at each of these intronic IPA sites, every time you lose another transcript, you cumulatively deplete the full-length messages. And this leads to really a downregulation of the homologous repair machinery. The phenotype that CDK12 exhibits is known as BRCAness because it phenocopies BRCA1 and BRCA2 mutations. And so these mutations are common in many types of cancer. CDK12 biallelic loss of function mutations are particularly frequent in both ovarian and prostate cancers. And so this plays an important role in tumor progression. And so we were, in fact, able to identify IPA sites using Cancer Genome Atlas data that were dependent on the presence of CDK12 loss of function mutations to upregulate these intronic IPAs in important genes such as ATM. And you can see that this results in a loss of the full length and presumably the functional isoform of this gene. So in summary, we uncovered in this work this sort of role of CDK12 in controlling the balance or the competition between the splicing out of introns and the use of intronic poly-A sites within them. And this reads out to many different functional gene expression groups, not only DNA damage repair, but many others, including transcription machinery itself. So this phenotype or this mechanism has been observed also by others. The George group and has also seen this IPA upregulation. And in subsequent work since we published this, Shona Murphy's lab has shown that likely this effect of CDK12 on transcription comes through a reduction in elongation velocity and or processivity, resulting from dissociation of these important elongation factors when CDK12 activity is lost. And so presumably by slowing down the polymerase, that alters the kinetics of this spliceosome and CPA balance. Other work by Ricky Johnson's group has also shown similar defects and effects on 3' processing in both CDK12 and CDK13 analog-specific, inhibitor-specific alleles. So in that case, what they saw is that really reducing both CDK12 and 13 simultaneously has the most profound effect. And this indicates that in some sense, their function is at least partially redundant. But they also found that there were distinct effects from knocking out the activity of either one or the other. So that brings me to the CDK13 project that I'm going to talk about today. This comes from a collaboration that started with a really talented physician scientist, Megan Insko, who's a postdoctoral fellow in Lynne Zahn's lab. And this project has really involved a whole bunch of groups, some of which have been added even more recently than our last manuscript version. If you're interested in this work, an earlier version of the paper is published on bioRxives. So CDK13 is actually implicated in some congenital disorders. So mutations within the kinase domain cause this spectrum of both dysmorphism in facial features, intellectual development disorders, and cardiac morphology defects. So this clearly has an important role in development that is distinct from that of CDK12. So Megan originally became interested in this gene when she found that CDK13 mutations are relatively more frequent in melanomas compared to other tumor types. And in fact, what she found is that in the Cancer Genome Atlas cohort from melanoma, that many of these same mutations that were seen in the developmental disorder were also found in human tumors. And these tumors carried a significant survival burden when CDK13 was mutated or low. So using an in vitro kinase assay, it was also clear that these mutations that were found in the human tumors are kinase loss of function mutations. So a in vitro substrate consisting of the RNA Pol2 C-terminal domain can be phosphorylated by the wild type, but not by the mutant kinases. So Megan then turned to this really useful model system for studying melanoma, that's the zebrafish. So these stripes in the mature zebrafish are actually comprised of melanocytes. And so these, a melanoma model can be devised by introducing an oncogenic BRAF mutation as driven by a melanocyte-specific promoter, as well as a deletion of p53. And so so eventually then this results in the formation of melanomas. So what Megan found here is that compared to fish with just EGFP expression, in addition to these oncogenic mutations, she could express wild type CDK13, the human wild type CDK13, or many of these human cancer associated mutations. And she found that these cancer associated mutations significantly accelerated the development and the progression of these melanomas. And in fact, the survival of the fish was severely compromised. And these cells, these melanocytes showed higher proliferation rates. So what we found when we looked at the molecular level, both in zebrafish melanomas from the model system I just described, as well as patient melanoma data from the Cancer Genome Atlas, and then in mouse ES cells, so Sarah Dubry, in addition to knocking out CDK12 in those cells, made a CDK13 deletion. And in all cases, what we found is that there was this gradient in polyadenylated mRNA sequencing data from the 5' end of the transcript to the 3' end, suggesting that these were being terminated somewhere in the middle and that these terminated messages were polyadenylated. We also found that after 48 or 72 hours of CDK13 depletion, the amount of reads mapping to the distal poly-A sites, the full-length isoforms, compared to the total for the gene, were also decreased, again, indicating early termination events. And we found using 3' end sequencing that these early terminations were predominantly found in introns, just like in CDK12. However, they seem to affect a different subset, partially overlapping but largely distinct, of intronic poly-A sites. And this is an example of one here from a human melanoma cell line with overexpressed CDK13 mutation, showing that we see an upregulation of this intronic poly-A site in the first intron. It has a canonical poly-A sequence upstream, and that results in a decrease in the full-length p53 message. And we saw that overall, both in the zebrafish melanomas and in the human cell line, that the predominant termination site was in introns. So interestingly, it turns out that these prematurely terminated transcripts can actually be exported and translated. And so using mass spec data, the peptides that were identified from these proteins in the presence of overexpressed mutant CDK13 showed this gradient similar to the RNA-seq data from the N-terminus to the C-terminus of the protein, suggesting that these proteins are undergoing C-terminal deletion. And here are two examples of individual proteins showing the distribution of peptides across their lengths. So what we did was to use an in silico translation to look at these intronic polyA sites globally. So if we take any of these events that have an IPA site in the intron, and we translate continuing from the upstream exon through the region of the intron that would be included in this transcript, we can predict the expressed peptides here. And so interestingly, many of these, the vast majority of these are between about 10 and 50 amino acids in length. And in fact, we were able to predict that many of these were high affinity binders for the HLA molecules present in the cell line in which these were detected. So, so this is just a schematic of showing again what we're looking at here. So if we look for the peptides that are encoded in this region of an intron, when there's an IPA site here, we predicted that perhaps we could find these in the mass spec data that was already performed. Now, these wouldn't be present in the normal databases to identify the peptides, so we had to use our in silico translations. But sure enough, we were able to identify a number of these intronically encoded peptides. These are from the zebrafish data, and these were entirely specific to the CDK13 mutation expressing melanocytes or melanomas. So to look at the molecular basis of this, Megan then did some co-IP experiments, and interestingly, she found that wild-type CDK13 interacts with a number of factors involved in RNA surveillance and other RNA stability. So first, she found that cyclin T1, which is actually the CDK9 cyclin, is the binding partner, at least in these cells, for CDK13. So previously, it's been found that cyclin K is the dominant cyclin, but in this case, it's cyclin T1. She also found interaction with an E3 ubiquitin ligase complex that's been implicated in elongation efficiency. Interestingly, NCBP3 is part of an alternative cap binding complex that's integral to sulfate, I'll talk about in a minute. And then also components of this PAX complex that targets prematurely terminated transcripts for degradation in the nucleus. So in fact, Lynne Maquat here at U of R just published literally like a month ago this review on the role of NCBP3. This has been well characterized by Torben Jensen's group, who also discovered the PAX complex. And to make a long story short, it seems that NCBP3 as an alternative cap binding complex is in competition with this subunit that recruits messages for decay by the nuclear exosome. So essentially, this is at the center of determining whether a message is exported for translation or degraded in the nucleus. So Megan then looked at, okay, so those were the wild type CDK13. What about the mutant? So looking at a particular mutation in the substrate binding pocket, she found that most interactions were unperturbed, including NCBP3, but this one factor, ZC3H14, was basically completely lost in the presence of the mutation. Now, whoops, and this is actually, sorry. No, I can't get it, so I'll just move to the next slide. So this is part of this PAX complex, ZC3H14. again shown by Torben Jensen's group, to be a subunit of some of these, perhaps substoichiometric unit of this complex. And what Megan found was that, in fact, when you lose CDK13, there's a loss of a phosphorylation site, S475. And so she then performed these pull downs and IP mass spec experiments using either wild type ZC3H14 and looking at other components of this PAX complex, or the nuclear export complex. And in the presence of the CDK13 mutation, the wild type ZC3H14 lost association with a number of these factors. But when she placed a phosphomimetic version of this protein in with the CDK13 mutation, this again restored the interaction with these factors. So this is my last slide here. So this brings us to sort of our overall model for both function of both CDK12 and CDK13. So normally what we think these factors are really doing is, in the case of CDK12, both suppressing intronic polyA sites, probably through its effect on transcriptional elongation. And then CDK13, among other things, also probably having some redundancy in transcription with CDK12, is also recruiting this PAX complex to degrade prematurely terminated messages. In the case of a tumor cell, whether it has a mutation in CDK12 that leads to increased IPA usage, or in CDK13 that by not recruiting the nuclear exosome stabilizes these truncated messages, perhaps through a competition with NCBP3, which then results in export and translation of C-terminally deleted proteins. Those we don't know, this is speculation, but perhaps those have loss of tumor suppressor function, or function as dominant negative tumor suppressor proteins. Or there could be a general RNA or unfolded protein stress generated by this. And then the interesting question then is, do these produce what could become a very tumor-specific set of neoantigens? So I just want to thank, first of all, you for listening today. I apologize for the glitch in connection. In my lab, Stas Belousov did the 3' end sequencing work and all of the necessary data analysis and scripting to do that work. Sarah Dubbery from Phil's lab, as I mentioned, worked on both the CDK12 and 13 mouse ES cells. And of course, I want to thank Megan and Lynne for having the opportunity to work on this really interesting project. And I'll take any questions.
Thanks, Paul. Now let's see, some questions are coming in the question answer. I want to remind everyone that you can use the raise your hand feature and we can call on you to ask your question out loud. The first question I want to ask is from Francois Bacand, and says, very interesting. Does deficiencies in ZC3H14 cause similar increase in IPA usage as CDK13 depletion? Your model would predict that?
Yes, so actually that's an interesting question. We haven't specifically tested that yet, but we have identified additional mutations within the cancer genome atlas cohorts in other factors that are part of this complex. And so some of the current ongoing work is actually testing whether those factors through 3' end sequencing have similar phenotypes. But the presence of those mutations potentially expands the number of tumors that have this type of RNA processing or surveillance dysfunction to a much larger set than the CDK13 ones alone.
Okay, so the next question came in the question answer from Sarah Dubbery. It says, great talk. Given the neoantigen observation in the CDK13 mutants, is there any evidence that patients with CDK13 mutant tumors would benefit from immunotherapy? And if so, could it be used as a biomarker for something like PD-L1?
Thanks, Sarah. Yes. So the answer is yes. That is a quite a potential both biomarker and therapeutic target. One of the problems with studying these mutations is that the overall frequency within any given cohort is relatively small. This is not like a p53 mutation. So I think the challenge there is getting enough patients who would then undergo immunotherapy and to have a statistical chance of determining whether that's effective. But it's a good idea.
Okay, the next question is one that can be posed by Nick Rugalis, who raised his hand. Nick, do you want to go ahead and unmute yourself and ask your question?
Yeah. Hi, Paul. Great talk. I just wanted to ask. I was thinking about it. Because when you were talking about CDK12, you also talked about it in terms of like transcriptional elongation. I was curious, like in terms of that and chromatin organization, have you looked at like any of like the histone proteins specifically, and like looking at whether, like if you looked at different variants or looking at different, like in the histone core octamer, like if you looked at any different mutations that like would affect potentially CDK12's function in the elongation process?
So thanks for the question, Nick. So we have not done that. There are certainly histone modifications that are associated with transcriptional elongation, H3K36 trimethylation, for example. And in fact, the H2B monoubiquitination that's carried out by the two of the cofactors that interact with CDK13 is also associated with dynamic transcriptional elongation. But we have certainly not studied that. And in the field, no one has really delved particularly deeply into that question, but it's a good idea.
Thank you.
So I think we'll take one more question. So Niels Walter asked, could liquid-liquid phase separation of the transcription and or RNA processing machineries based on phosphorylation status play a role?
Absolutely, yes. This is likely to be a phenomenon that contributes to all of these processes. It's certainly been shown that transcription is associated with liquid-liquid phase transitions, and as well as, at least in plants, there has been some evidence that the cleavage and polyadenylation machinery is also partitioned. So I think the answer is absolutely yes, but we don't know specifically how that works yet.
So maybe we'll take the one last question that came in from Peng Yao. What is the level of the IPA-derived truncated proteins at baseline? Are there any beneficial functions for those truncated proteins?
Yes, so we think they are very, very low. So all of these quantitative assays are done in a relative sense, of course. So when we see this increase in these intronically encoded peptides in the presence of the mutation, we're looking at log fold change compared to the wild type. But our hypothesis would really be that those are not typically present at high levels under steady state conditions because they're being suppressed by this very machinery. So we hypothesize that CDK12 is primarily suppressing the generation of these truncated messages, whereas CDK13 is basically cleaning up anything that makes it past CDK12, causing it to be degraded before it can be exported. Because we don't understand how these CDK13 mutations ultimately accelerate melanomagenesis, but presumably it's due to the production of these truncated messages. So presumably these would be things that the cell would want to suppress.
Thanks, Paul. And thanks, Clara, and thanks, Liz, for all your help organizing today. And thanks, everybody, for joining us. The seminar will be posted to YouTube for future viewing. Next time, there'll be 2 speakers from Harvard Medical School, Sunit Agarwal and Carmelo Nucera. And I hope you'll be able to tune in and join then on November 18th. And goodbye for now.